Oh, I have a few familiar names in the crowd. Good morning, everyone. Let's just wait for everyone to connect.
Good morning.
Morning.
Good morning.
Morning.
Hi.
Looking good. Okay, lovely. Well, again, there's so many people that I can't see you all. So if you have questions, write them in the chat or raise your hand and I'll try to keep track of it. Other than that, the rules of the game are we meet every Monday morning. As you all know, you're here. You can, nice glasses, Leonardo.
Good morning, everybody.
Morning.
That completely threw me. Every Monday morning, 9:30. It lasts maximum an hour, depending on how many papers we have. Everyone is welcome to bring a paper and discuss it. And everyone is welcome to ask questions. Chances are, if you didn't understand it, someone else didn't. So just ask and we try collectively to answer your questions. With that, I'm aware of 3 papers today. Is there anyone else who wants to present a paper? Let me just see the chat. Jesus, I have Michelle and Leonardo right next to each other doing the pirate thing. Okay, right, with that, I'm going to hand over to Laura, who's going to present the first paper. Laura, if you can share your screen. Everyone else, you're welcome to keep your videos on. It's nice to see people in the room, but we would ask you to kindly mute yourselves while someone is presenting. Thank you.
Okay, do you see it? Okay, so this paper came out kind of 10 days ago in Cerebral Cortex, and it is a study which takes into account a specific population, which is the one of people affected by agenesis of the corpus callosum. Basically, the authors tried to assess the structural and functional connectivity in this group, and they also led a correlational analysis between these connectivity patterns and the neurobehavioral outcomes. What they hypothesize is that since we have a reduction of interhemispheric connectivity, then we could have a strengthening of intrahemispheric connectivity. And they also hypothesize that different patterns, different degrees of strengthening of this intrahemispheric connection could be related to different outcomes for the behavioral part. So basically, what the authors did was to take this data from a dataset, which is the pediatric agenesis of the corpus callosum project. And it is a database which contains structural information, so structural MRI, diffusion MRI, as well as resting state fMRI, and also cognitive testing. So what they did was to compare these data coming from the agenesis of the corpus callosum population, so children with this condition, and control groups of typically developing children. So this is the flowchart of the analysis that they carried out. They considered functional MRI data, and then they computed some functional connectivity matrices by considering the time series of the activation of different brain regions. These brain regions were, let's say, the brain was parcellated according to this atlas, which is the brain ectome atlas. So they analyzed how different regions from this atlas were activated together and at the same frequency. And then for the diffusion MRI data, they computed tractography. They, let's say, segmented the tractography that they did with this atlas by doing a pairwise association, pairwise segmentation, let's say, of the whole brain tractography that they had. So they computed the structural connectivity matrices for 2 main measures, which are the fractional anisotropy measures and the number of streamlines, which was weighted for the volume of the regions of interest of the pair. So let's say, on the other hand, for the behavioral part, they had data for general cognitive functioning, short-term and working memory, executive and attentional functions, learning and memory, and the social functions. So what they got for the connectivity part, as you can see here, they carried out the analysis at two different levels. So we have the whole brain level, considering left and right hemisphere, and then we have the lobar level, so considering the different lobes of the two hemispheres. This is the row for the agenesis of the corpus callosum group, and then the control groups in the bottom row. For the structural connectivity, they noticed that there was, of course, a reduction of interhemispheric connectivity comparing the two groups. They also noticed that at the lobar level, we had an increase of structural connectivity, in these cases for the fractional anisotropy measures. And this is true for the frontal, the temporal, and the parietal lobes. As you can see here, the intrahemispheric connectivity occupies less space in the typically developing controls compared to the agenesis of the corpus callosum group. This is not true for the occipital lobe, in which you see there is the same entity of this measure. They basically postulated that the lack of interhemispheric connectivity would lead to an increase of intrahemispheric connectivity. In this case, it's fractional anisotropy, so it would mean, I don't know, better organization, better myelination of the fibers. On the other hand, if we consider the functional connectivity, there was no significant difference between the two groups. So we might say that basically the changes that we have at the structural level, the compensation that we have with the increase of the strength of intrahemispheric connection would allow also the patient's group to have a normal pattern of functional connectivity. If we consider the correlation between the connectivity and the behavioral measures, basically they did a partial least square analysis. They computed the Z-score for both the connectivity measures and the behavioral testing results. And they did this singular value decomposition, which allowed them to have to extract some latent components, so that each latent component was made of a pattern of connectivity. In this case, it's structural, as you can see here, fractional anisotropy and number of streamlines. And this same pattern of connectivity for the two groups was related to different outcomes in the behavioral part for the agenesis of the corpus callosum and the control group. So they consider this component so that, let's say, strengthening a better, a stronger, let's say, intrahemispheric connectivity for the fractional anisotropy and the number of streamlines measures, and a lower, worse, I don't know how to say, connectivity for the interhemispheric part would be related to specific patterns of behavioral performance. In this case, we see that there are some tests for which this pattern of connectivity was related to a better performance in the two groups, which are, for example, we can see here the dual task, then we have the working memory. Then we also had some divided attention. Here we have other divided attention tasks for multimodal, for different modalities. It is, let's say, nice to notice that in the agenesis of the corpus callosum group, in general, this pattern of connectivity would lead to a better performance for executive functions and attention in general, as well as verbal learning and memory, it is, yeah. So on the other hand, we see that when we have this pattern of connectivity, we have a worse performance in the control group, in the batteries for verbal learning and memory, for these tasks of executive and attentional functions, and also for these tests for general cognitive functioning. Then the last part is the study of the relationship between the functional connectivity and the same cognitive skills. In general, when we have a stronger functional connectivity, both intrahemispheric and interhemispheric, in both groups, we have an overall better performance for executive and attentional functions. Specifically to the agenesis of the corpus callosum group, we have a worse performance in this task, which is the verbal immediate recall task. And interestingly, we have different patterns of association for the dual task in the two groups. So that if we have a stronger functional connectivity in both intrahemispheric and interhemispheric, we have a better performance for the dual task in the agenesis of the corpus callosum group. And on the other hand, if we would have a lower functional connectivity for both intra and interhemispheric, we would have a better performance for the dual task in the control group. So basically, this is what they found, and it is in line with the hypothesis that they did. I think it's very interesting the fact that they, in the discussion part, they did an analysis of the, let's say, neurodevelopmental steps that we have even during gestation, and which can lead to different patterns of agenesis of the corpus callosum. They also corrected for these different patterns that we can have, so partial agenesis or other things. And they also proposed that, for example, given that we have some functional connectivity that is the same in the two groups, maybe it can be also supported by other type of commissural connection, such as the anterior commissure. So that's it. If you have any question, I'll try to answer.
Thank you, Laura. Any questions in the room? Okay, Michelle, start us off.
So it's for like the connections that will allow the interhemispheric exchange, because they use those regions in the left and in the right hemisphere. If that was the entire commissure, they should have seen on the structural connectivity matrix this kind of connections between the temporal lobes, and they don't. So maybe it's more like thalamic, or I don't know. Do they use subcortical?
No, as far as I know, no. But this is something that I couldn't explain to myself because I was wondering if we have functional connectivity. Part of functional connectivity is subserved by intrahemispheric connections, and part of it also depends on interhemispheric connections. So even though we have like a balancing with a strengthening of the intra, how can we have the same pattern of functional connectivity in the two groups when we lack like a big part of interhemispheric connectivity?
I mean,
and yeah, go ahead.
Sorry. So we've been working with Steffi on agenesis of the corpus callosum, and usually we see they either have a part of that corpus callosum that is preserved, and here I think we just see like an average, so it's difficult to really see whether or not they were all having a full absence of the corpus callosum, or a really big entire commissure, but here we don't see the connection between the temporal lobes. And then like the other big candidate after that will be the interthalamic commissure. So, you know, also brain project on the thalamus, and the thalamus is extremely connected with himself, with the other hemisphere. But since they didn't have subcortical region of interest, it's really hard to to see whether this was true or not.
Okay, just one thing for the stuff of maybe they had only a partial agenesis or this, let's say this part of the discussion. They also did an analysis of the structural and functional connectivity, whether they differed between people who had the complete agenesis and people who had only the partial, or maybe if they had the agenesis, but also accompanied by other conditions and everything. So it shouldn't be this, it should be control for this. But yeah, I agree with you for the other types of connection.
That's an amazing paper anyway. That was very well presented. Thank you.
I would just follow up and show you what the figure that Michelle was talking about. I'm going to stop you sharing your screen for a second and just show you this figure. Right, so this is from our 2014 paper where we looked at agenesis of the corpus callosum. And you have the healthy brain anatomy here with a really strong corpus callosum in red. And then you see the different degrees of agenesis in 3 subjects here. And it is a well-established fact that if you lose the interhemispheric connectivity through the corpus callosum, these fibers still develop, but they stay within one hemisphere, so they become intrahemispheric. And that's what we see here. So this is the whole debate about the superior frontooccipital fasciculus that may or may not be part of the interhemispheric connections of the corpus callosum. And this is what we can see. And obviously, if you have less interhemispheric connections, but more intrahemispheric connections, you have less crossing. So I would expect the FA to go up in that case. So that's not a big surprise, personally, I think. That's just as a comment. And then one thing I may have missed, trying to multitask here, is how they found the agenesis, because they're usually subclinical and are incidental findings. So how did they actually find the participants for the study?
Basically, I mean, they use this data set, but the first author of the paper, this paper is also the first author for the paper presenting the data set. And they explain that they consider these people who had a diagnosis, a previous diagnosis of agenesis of the corpus callosum. And they also involved an expert that I don't know which type of figure does this, but basically they ran scans on these children, and this person confirmed the agenesis and also the entity and the type of agenesis that they had.
Right, okay.
I don't know.
Thank you. It's a really great paper. Thanks for presenting it. Just before I throw the mic back to the audience, for those watching us on YouTube, I have a second screen up. So if you want to post your questions there, we can pick them up and throw them into the Zoom mix. So just let you know. Patrick, you had a question.
Yeah, thanks for presenting, Laura. I was just wondering, I don't know if there's any paper that actually shows something like a temporal to temporal connection in agenesis patients. Do you guys know of anything like that? Because I just remember the Tova Moll paper 2014 that shows like an increased parietal connectivity due to not increased but compensatory parietal connectivity via the anterior commissure, but I don't think that there's anything showing an interaction between the temporal lobes, so that there shouldn't be in agenesis patients.
I guess it depends on the degree of agenesis, to be honest. As we've just seen, there can be partial connections missing or the whole structure.
Yeah, but what I mean is, in general, like, is there anything known about temporal connectivity through the anterior commissure? Because as far as I know, the anterior commissure, for instance, or the other commissures shouldn't be able to reach that far.
Can I say one thing?
Sure.
I don't know about the temporal, but in this paper, they say that they found in people with agenesis of the corpus callosum that, I don't know if this is, I don't know how to take this, but I say it. The fact that we didn't have an increase of intrahemispheric connectivity for the occipital lobe is due to the fact that some interhemispheric connectivity might be preserved through the anterior commissure. This is what they say. I don't know how to interpret this because it doesn't seem to me very ecological.
We're talking occipital lobe, but anterior commissure?
Yeah, that's, I had the same reaction. I was like, okay, this is not very,
so like that, you know, some description of the anterior commissure show connections to the occipital lobe. Whether it exists or not is discussed, but you know, this is really, you know, some people saying that's because there've been some mix-up with optic radiations. Some other things that actually you truly have those connections of the anterior commissure reaching out to the occipital lobe.
But regardless of whether or not that is an anatomical entity that the anterior commissure is projecting to the occipital lobe, but with the missing splenium of the corpus callosum, you would expect to see a change in connectivity between the two occipital lobes. That's still quite a strong structure, isn't it?
I remember looking at Agenesis of the corpus callosum for the first time with diffusion and being, I am in an alien brain, like really doesn't look like a classical human brain, if I can say that. It's really surprising the way like the white matter reorganized itself.
When you lose all your anatomical landmarks with the absence of the corpus callosum.
Just one further remark. Were there anything like an increased intrahemispheric connectivity shown in the Agenesis group due to the props bundles or something like that?
I don't know.
Well, that is part of the whole debate about the props bundle and the superior fronto-occipital fasciculus. So you can definitely find papers, but take them with a grain of salt and read the methods carefully. Okay, I can see everyone muted themselves. I take that as no further questions. So with that, I'm going to hand over to Victor to present the next paper. And I'm going to leave you guys alone for a second because I need to plug in and I forgot the plug downstairs. So Victor, if you can start your screen sharing, I just make sure it works and then I'll be back in a sec.
Okay, is it okay for everyone?
Yes.
Okay, perfect. So this was an article published, I think last week in Nature Neuroscience. I must admit that I haven't had the time to completely read it. So it will be more of an introduction to the article from what I've read, rather than a complete presentation. But I think it's a very interesting article, and it touches a lot of different fields that are interesting when you start to learn about functional connectivity. So this article is he's studying the functional connectivity of the human subcortex. So basically the gray matter nuclei of the human brain. because it's a region that is not that well explored in functional connectivity. Usually there are a lot of studies about the cortical gray matter functional connectivity. There are some, of course, that are focused also on the subcortex, but it's not as well explored. So what they did was so just to go back to the beginning, there are several different approaches to the study of the areas, the functional areas in the brain. You can either, well, the two main way to study these areas is either to create some kind of cortical boundaries, where you say, okay, this is one area that has one type of functional signal inside, and it will be used for this and that, and it is part of it is part of a resting state network, and it is like well delineated. And there is also the opponents, well, kind of opponents, they say that we should not create like boundaries, but it's more of a gradient when we switch from one areas to another, but gradually, so we cannot have limits, strong limits between them. And so it's true that it's still an open debate in the field, especially for the cortical signal. But here they wanted to focus on the subcortex, and they used the gradient method. So really, how the functional connectivity changes over the regions in a continuous manner. And they created a new visualization and analysis method that they call gradientography. So what they do, basically, if I go to the figure here, so they, for each voxel of the gray matter nuclei, they compute the functional connectivity fingerprint of each voxel with the whole brain and with the subcortex, and they do they do a PCA to get the gradients of the different fingerprints, to know where are basically how are the voxel related between each other, and where are the gradients. It's pretty hard to explain on the go for this kind of thing. So I really encourage you to look into it. But so basically, it will create a map. So we can see here a map of of one type of gradient. The first gradient here. It's hard to explain, but you can see here the map corresponding to the first gradient here, which is one type of variation that you can find in the connectivity matrix, and it is projected on the gray matter nuclei, showing the direction of the gradient of variation of connectivity. I really have a hard time explaining that, but basically it will show where the direction of the changes in functional connectivity across the region. And so depending if there is like a large variation of functional connectivity, you could usually say, okay, this is there is a changes of type of function of connectivity. So we have a different functional region. So what they do here is have on this map, they have the vector indicating the direction of the gradient. So basically, this gradient goes from high to low in this direction. So you have like a vector showing this direction, and then you have this for every voxel. So what they do is to use a tractography algorithm to link each voxel together to create some kind of streamline going from one part of the side of the gray matter nuclei to the other, going all the way. This is the right images here, and so it really englobes the whole thing. Well, they separate it into dorsal and ventral path streamlines here, and so what they do then is to map on the streamlines the value of the intensity of the gradient. So basically, when you have a high value gradient, they will put a different color, and you can think as of it as kind of a way to delineate the different parts of the streamline, where the streamline finds a boundary. And to really know where they have a boundary, they took all the streamlines and they summed that. Well, they did the mean of their values across the length. So you have the lengths here on the x-axis, and the value of the and the value of the vector in the y-axis. And then they looked for the local maxima of these mean, and these give them the putative boundaries between each functional region. And they did it so for the dorsal and for the ventral part of the gray matter nuclei. Well, it seems to work really well. I think it's so. Afterward, they do also a whole atlas, and they do replications, replication studies, and everything. So it's very comprehensive. But the method, when you start to be when you are familiar with the method, it's an interesting take on functional resting state connectivity, but I'm not completely sure, because I still have to read it entirely, but I'm not completely sure how they managed to have streamlines going, all the streamlines going from one side to the other, and to plot them like they did in the on this graph. But it's a really interesting approach to the delineation of a functional domain inside of the gray matter nuclei. Plus, it's a good take on this, because this is something that is not explored enough in the field. So, well, I hope it was not too confusing, but I really encourage you to read it, and at least to start on this one for those that don't really know about it, and to explore the all that they talk about, because it's really it's really a take on very many fields in the with this approach. So you really have a quite a comprehensive view of the field when once you've read it and you've looked into the different things that they talk about.
Thank you, Victor. Definitely sounds like not the easiest paper to disentangle. Any questions in the room?
No. May I start?
Yes,
I feel like
Patrick, go.
So just thanks again for presenting, but just to clear some things up, because I'm kind of lost here. So, and probably others are too. So they used first functional images, right?
Yep.
To get the fingerprint for each voxel in their subcortical areas.
Yes.
And they delineated 3 gradients based on the Laplacian eigenvector map.
Exactly. Yeah.
Basically, is an approach to get like a dimension reduction of these complicated fingerprints, right?
Mm-hmm.
And then it looks like they used kind of a simple like diffusion tensor.
Exactly. Yeah.
To, right?
That's right.
Okay. Okay. But I kind of don't get what is then shown in the very right image that you're having on your screen right now, where it shows gradient magnitude. Is that some like akin to fractional anisotropy in diffusion images? Or
Can I comment on this? So because I know Daniel, I know the theory that is behind it, and the idea is like gradient, with evolution, they left an imprint in areas that like is decreasing on as you're going further on the evolutionary tree. So by looking at the directionality of this progressive decrease of the gradient, behind that you can infer like how different cells have been like growing and multiplying with evolution. And that, I think that's what they're trying to show here.
yeah, but if that would be like what they just trying to show, then wouldn't. So what I'm wondering is the gradient magnitude. Is there basically an additional step to just showing the gradient on top of the brain map?
Here it's to delineate the different functional domains inside of the for example, of one streamline. If you take one streamline that goes from one side of the whole nuclei, and then you just unravel it all the way through, and you have like different parts where you have like as we can see in the right part, the right figure, you have different value for the of the vector, and so it's really like on this image, like you can imagine that this is one streamline, and you have like different peak and troughs, and this is just like when in visualization where you see everything, so you can get a general idea of how it works. But I think it's more interesting to see once you've plotted in 2D to really get an idea of how they delineate stuff here.
Can I add or try to add something?
Sure.
Go for it, Leonardo.
Something like 2 years ago, they published a paper, well, Daniel and some other people, they published a paper on scientific communication where they looked at different gradients of functional connectivity in the whole brain for people with autism and without. And one similar plot to the 2D plot that you see here was quantifying the ease of the rate of transition from one point in the gradient to the next one. Mm-hmm. So what I think is being shown here is how fast is the transition along the gradient. Let's say if you have regions in which the transition from one part of the gradient to the other one is completely uniform, well, not uniform, yeah, uniform actually, then you will have a very continuous color, but solid. But if you have parts of the gradient in which the transition of the information, let's say that there is information flowing along the gradient. If you have parts of the gradient in which the transition is more difficult or faster, let's say in one group or in the other group, then in this case, you will have these hills. As a matter of fact, in that paper, there were quantifying differences in rate of transition of the information between primary sensory and transmotor cortices. And I think that here the idea of what they are representing is exactly the same. So you don't only represent the gradient itself in the gradient magnitude, but you also represent how fast is the transition from the previous to the next point of the gradient.
Mm-hmm. Cool, thanks.
And that, as a matter of fact, can be indeed an indication of the segregation between two different parts of the gradient. Because if you have you can think about having very quick transition within a certain functional unit and to have slower transition between one functional unit and the next one.
Okay.
Okay. Are there any other questions or comments on this paper at the moment? Doesn't look like it. Okay, so we move on to the last paper for today, which will be presented by Lia. Can't see. Ah, there you are. Hi.
Here I am. So start the sharing. Okay, you can see the screen, right? Yes. Changing the subject a little bit, here this paper published online on the 26th of October by Remika Mito et al. In Neuroimaging Clinical, and it's talk about white matter alterations that you can have in white matter lesions that usually are detected using FLAIR images, but I can show you an image. So in the FLAIR image, you can have these lesions that are present both in people who show the dementia, like Alzheimer's disease, but also in elderly people. Indeed, in this study, they compare a cohort of both healthy and healthy controls, 94, and patients with Alzheimer's disease, 38. And in the study, they also use diffusion imaging to evaluate these lesions and not only FLAIR images. Because they want to see if in these lesions they can detect different characteristics based on diffusion imaging. And to do so, they used an approach called single shell 3-tissue constrained spherical deconvolution. So they evaluate the microstructural organization using a constrained spherical deconvolution, which allow you to describe how the gray matter is organized, in terms of corrosing fibers, and they also and diffusivity profiles, and they used a thing that I found interesting, a single shell acquisition. So using only one V value, in this case 3000, because I saw this 3-tissue evaluation. So starting from the diffusion signal, you you are able to distinguish 3 classes that here are called white matter-like, gray matter-like, and CSF-like tissue composition. And usually I saw that this approach was possible using multi-shell data, and in particular also lower p-value. But in a generalization of this approach, you can also use a single shell acquisition that is useful when you are trying to translate the advanced neuroimaging analysis on the clinical practice. To have a look to the methods, so they evaluated flare images, and they used actually an automatic segmentation of white matter lesions. These are these hyperintensities. In the clinical practice, this is really debated because usually you don't know if some lesions are pathological or not, because we have said they're also present in healthy controls. But they segmented all these lesions automatically, starting from the flare image, and then they evaluated. They divided this hyperintensity in two ways, based on what is currently done in the literature and in the clinical practice. So they divided the periventricular lesions next to the ventricles, and the deep white matter lesions that you can see here in red. And this was one classification. In the other classification, they divided the hyperintensities based on the distance from the ventricles, starting from the one who are more close to the ventricles, and so region 1, then you have region 2 and region 3. Here you have also the distance. So region 1 within 10 millimeters, then region 2 20 millimeters, and then region 3 in red. And here, if in the evaluation of periventricular and deep white matter lesions, if the lesions were confluent, you can use a threshold of distance from the ventricles that is 20 millimeters. They create this data set of lesion segmentations using these two different approaches, and then they evaluated how was the diffusivity profiles based on the three different categories. So white matter-like that you have in blue, gray matter-like that you have in green, and CSF-like. And here you can see the different maps. Indeed, what they show, this is a patient. So to explain that here in the segmentation of the white matter lesions, you have a different composition. So you don't only have one categories, but there is a mix. And in this article, they try to describe this composition in white matter lesions. Indeed, if you divide, so as divided lesions, periventricular white matter hyperintensity, deep white matter hyperintensity, and also normal appearing white matter, because this is a point of debate, because if you segmented the T1 images, and they use the FAST for this, FSL tool, SPM tool, no, FSL tool, sorry. And they evaluate also the composition in what it seems a normal appear white matter. And as you can see in both the groups, you have a similar composition in Alzheimer's disease patients, lesions and healthy controls. And here you can see that periventricularly, you have the three categories that are quite close. Instead, on the normal appearing white matter, you have a relative Sigma fraction that is like higher, so more white matter-like. And then you can also show this composition using a ternary plot. So here you can see where the normal appearing white matter is really different from the composition Here we have the subdivision of periventricular and deep lesions that they seems to intersect here, but you can see a difference in the two groups. And also, if you divide it in the three regions according to the distance to the ventricles, you can see again that the normal pure white matter has a different composition, and also the region 1, so next to the ventricles. Instead, the region 2 and region 3 are quite similar in the compositions. And also in the supplementary materials, this is an explanation about the ternary space. So you have an isotropy on one axis, diffusivity in the other, and the three vertices that are white matter-like, gray matter-like, and CSF-like. Other examples of these different composition of white matter lesions, and also the topology of the lesions. And here I wanted to show you that actually if you divided the two groups, so Alzheimer's disease patients and healthy controls, you have quite an overlap between the two in terms of microstructural composition. Instead, if you compare the two groups, looking to the extent, so the volumes of these lesions, you find some differences as we expect and know from the literature. In particular, of course, in the Alzheimer's disease group, you have a higher volume evaluated white matter hyperintensities, and this is a significant difference. And if you split the lesions in periventricular and deep, both the volume of both the lesions categories are significantly different. But interestingly, if you divide it in the three regions, you don't find any difference in region 1 and region 2, so the ones that are closer to the ventricles, but you found a difference in the volume in the third region, so going more deep in the white matter. And indeed, I find that this is an open question to evaluate these results in terms of the translation to the clinics, and also to give a significant a meaning of these lesions that are also present in healthy people, because it seems that the periventricular lesions are the one who are more related to cognitive impairment and dementia. But as shown in this study, they are also present in healthy controls, so they seem also more benign, so more reflecting a normal aging. And as they show, the microstructure organization is also close in the two groups. And looking to histopathological studies, post-mortem studies, you can find different patterns of these. They show this in the introduction. You can find like different profiles in the histopathological evaluation, including like alteration in the myelin, gliosis, and the most common white matter infarction, so something that is related to a small vessel disease. And so I found this article really interesting, and I like that the clinical translation, but yes, I think that there are a lot of open questions according to this field, and how to characterize the lesions and how to really assess the correlation to the clinics in patients, but also healthy controls. So if you have any questions, I try to answer.
Thank you, Lia, for this paper. Shomisha is happy to see more triangles in there.
Yes, I didn't want to say that, but there are triangles.
Oh, he loves it. It's very interesting, that paper. I don't have a particular question as such at the moment, but like it's one of the holy grails to disentangle the lesion compartments and the different tissues and different types of pathologies within one lesion. So this is quite an interesting paper to look at. Are there any questions in the room? Doesn't seem like it. Okay, well, with that, that was the last paper we had for today. Thank you all for coming. Just to let you know, this week we have a CNS seminar special on Thursday and Friday from 4 to 6 p.m. It's the CNS Comms, where we teamed up with the Springer Nature Network and in particular Brain Structure and Function, and we'll present their special issue where all the authors who submitted and published their paper in the special issue will present their papers and what they're about, why they're important, what they did, how they did it, all within 10 minutes. So you get a really nice snapshot of the current updates of the structural connectivity of the cortex. So this is happening this week. Again, it's on Zoom and YouTube, so just click the link on the website and come and join us, or watch it afterwards on YouTube. That's it for today. Thanks for coming. Have an exciting week in science, and we see you next week. Bye.
Thank you very much.
Thank you.
Bye.
Bye. Thank you.
Bye.
Thank you. Bye.
Bye.
Thank you. Bye.
Ta-da. Hello.
Ciao.
Did you cut the live?
We are live.
We're still live.
Not
Still
anymore.
live. Cut it.
Done.
Sorry.